In this lecture, we're going to be focusing on adolescence and the aging brain. After we've established kind of connections and solidified them, the brain undergoes kind of a reorganization process during the adolescent period that unfortunately creates a situation where the executive functioning areas, which we'll talk about in this lecture, are less active compared to the activity of impulsivity and risky behavior. And so this period of life, the adolescent period of life, is characterized by increased risk and impulsivity without complete awareness of long-term or even immediate consequences. So let's get started. The adolescent human brain represents slow maturation of the human brain, especially those that are part of the executive function areas. So executive function refers to functions that are handled by the prefrontal cortex, things that involve decision-making, planning, weighing consequences, judgment. All of those are considered executive functioning, and they are handled by areas that mature the last. And so by maturation, we're talking about myelination of this area. So the prefrontal cortex is one of those areas that gets myelinated last. And so the efficiency or effectiveness of these areas of the brain aren't as good. And so they don't perform as well as they need to. And that's definitely characteristic of the adolescent period. So yeah, it results in the slow myelination of the prefrontal cortex. The consequence of this slow myelination of the prefrontal cortex, or slow maturation of the executive function areas, is that a person during the adolescent period has immature responses, such as impulsivity and risky decision-making, during this period of their life. Unfortunately, it creates the individual engaging in precarious behaviors, which may or may not lead to some deadly consequences, right? So something as simple as texting and driving may seem completely innocuous, but regular engagement in this behavior could put you in situations where you could get into a fatal car accident, right? So it can lead to deadly consequences. To study the kind of changes during the adolescent period compared to the adult and developmental brain, Dr. Luna and colleagues at University of Pittsburgh explored executive functions in children, adolescents, and adults. Specifically, they were trying to study something called response inhibition. Response inhibition is an element of executive functioning, and it requires us to inhibit any responses that might interfere with the completion of a task. So typically, when we have to engage in a task, we have to plan and then carry out that task, but we also have to ignore any irrelevant information or stimuli that may may interfere with or get in the way of completing that task. So that is response inhibition, inhibiting the response to pay attention to those irrelevant stimuli so that we can focus on a task. An example might be like if you're in a crowded party and you're having a conversation, but you hear somebody else call out your name, you need to inhibit your response to eavesdrop on the conversation where you heard your name and instead focus on the conversation that you're having at the party. That's response inhibition. So they created a study that was very similar to this social situation. And here's what they did. Participants were placed in an fMRI scanner, and while they were in the fMRI scanner, there was a light that they had to kind of stare at. While they were staring at the light, another light stimulus appeared off to the side. As human beings, our natural instinct would be to look at that new light that just appeared. That's visual reflexes, and it's something we have to purposefully ignore. And so what these participants had to do when that light appeared off to the side, not only did they not have to look towards the light, but they had to look in the opposite direction of the light. So the task was to look in the opposite direction of the light and inhibit the response to actually look at the new light that showed up, right? So it requires a fully functioning prefrontal cortex to engage in this type of behavior. So this type of control, where we not only ignore the stimulus that may pull our attention towards it, but continue engaging in the task, is called top-down processing, and it is a part of executive cognitive functioning. So here's what they found in the study. They found that over the three groups, children, teens, and adults, there was a gradual improvement in this task. With children, 50% of the kids had errors, whereas adults only had 15% of errors, demonstrating that adults have greater cognitive control. What's really interesting is that the teens had a pretty close error or correction in their inhibitory response compared to adults. But when they looked at the brains of the adolescents compared to adults, it looked like the teen brains had to work even harder compared to the adults because they were dealing with a brain that was not fully developed, or I should say a prefrontal cortex that's not fully developed. So this suggests that the teen brain, while it may be good or capable of executive functioning, has to work even harder to compensate for the fact that their brains are not fully developed. So teens often are driven by something called immediate gratification, this desire to kind of accept current rewards and forego rewards that may occur later. Part of the reason why they are into immediate gratification rather than delayed gratification, which is often what you see in adults, is because their brains are functioning differently. The ventral striatum, which handles processing of rewards, is more active in teens. And the prefrontal cortex, which kind of regulates our behaviors, is underactive. And the amygdala, which is important for avoiding harm, is also underactive. So the end result of the ventral striatum kind of driving the car or the brain is that the teen brain has a more enhanced attention for immediate rewards and would rather receive those immediate rewards rather than engage in what's called delayed gratification. Ultimately, the teen brain, when we're comparing kind of the changes in functional development over age, the limbic regions, which handle emotional processing, are more developed than the prefrontal cortex regions, which handle executive function. And this part here, outlined in purple, represents the risky period, and the adolescent age is like in the fattest part, where the limbic regions are almost completely developed, the ones that are seeking gratification, seeking emotional reward, immediate responses, and the prefrontal cortex that might be able to kind of inhibit those responses are not fully developed yet. And so that's why the adolescent period is the time of life where individuals often engage in risky behaviors because their brains have not fully developed the ability to weigh consequences of those behaviors. The other element of adolescence is kind of a risk associated with deteriorating mental health. So adolescence, if you know anything about mental disorders, is a time period where many mental disorders typically start. And that includes depression, schizophrenia, bipolar disorder, eating disorders, anxiety disorders, as well as substance abuse. So pretty much any mental disorder that's relatively common, even the ones that aren't super common, can start during the adolescent period. The adolescent period is the time when many of these mental disorders have their onsets. So scientists suggest that it might have to do with the fact that it's during this time period where a lot of brain maturation occurs, and there are dynamic changes occurring in the brain at this time. So any environmental challenges, things like stress or a dramatic life change that may impact brain development, puts the adolescent brain at risk for developing these disorders. So disorders aren't simply genetic, they are an interaction between the environment and genes. And so adolescence is a time period where this interaction between environment and genetic risk is quite high, leading to the development of many of these mental disorders. And so it might be the stage where development is so susceptible to mental disorders because any environmental challenge that exists may negatively impact brain development, making it more likely for the adolescent to develop the disorder. Once the adolescent period's done, the next kind of dynamic change that occurs is in the aging brain. So the adult brain is relatively stable until we get to about like 50 years of age. 50 years onward, actually, some people say as soon as the brain is done developing, it starts to deteriorate, which is not a good situation for me to be in, but we're going to take a look at the aging brain and the things that are typically found in a brain that is, from 50 years and onward. One thing I will highlight that is true of the adult and aging brain is this idea of neuroplasticity. Decades ago, scientists used to think that the adult brain was this rigid structure that once you kind of underwent development, that was it for changes occurring in the brain. We now know that the adult human brain is quite plastic. And I'm going to share with you some evidence to show this plasticity that exists in the adult brain. So the first evidence of neuroplasticity actually came from studying primates, specifically owl monkeys. These are owl monkeys that were studied at Johns Hopkins. What they did was they mapped out the hand, right? The thumb, first, pointer finger, middle finger, ring finger, and pinky finger onto the somatosensory cortex of the owl monkey. So they found where these digits mapped into the brain, and then they amputated specifically digit 3. Okay, so this is the middle finger of the owl monkey. They amputated it, but the monkey was able to function fine without this digit. After amputation, several months after amputation, they checked this map of the somatosensory cortex and found that, not surprisingly, the part that used to be handled by digit 3 was taken over by digit 2 and digit 4. So those parts that were kind of relegated for digit 3, rather than wasting that space away, was taken over by the surrounding 2 digits, making them more sensitive. This suggests that even the map of the body is capable of being adjusted and changed, demonstrating that the owl monkey was plastic. So this showed that, okay, if you remove a part of the body, can the brain change? Sure. And that was certainly what they found with this particular study. The next thing that researchers were curious about was, does the brain change with varied stimulation of different body parts? So let's say, there are soccer players, right? They use their legs way more than the typical typical person that doesn't play soccer, for example. Would the parts of their brains that controlled their legs be bigger in soccer players than than in people who didn't play soccer, right? So is there plasticity with stimulation rather than just removal? And so they did another study, still with owl monkeys, to illustrate that plasticity is not simply occurring in situations of tissue loss or body loss, but instead can also be present in situations where there's added stimulation. The brain can compensate by making brain areas larger. So in this particular study, they had owl monkeys. They measured owl monkeys before training. So they measured, again, mapped where in the somatosensory cortex their 5 digits existed. And then they had them undergo training that forced them to utilize digits 2, 3, and 4 compared to the other 2 digits. After months of training, what they found was that digits 2, 3, and 4 expanded their space in the brain to compensate for that added stimulation, suggesting that not only does the brain change with, the absence of a finger, but the added stimulation or activity of different body parts can contribute to increased space and modifications in the brain. Researchers have also shown this to be the case in human beings, especially musicians and violin players shown here. What they did was they put they measured the cortex of violinists in an MRI study and found, so if you're not aware, when you play the violin, your left hand is getting a lot of stimulation by playing these different strings and having to move a lot more. And they predicted, and they found exactly this, that the right hemisphere that handled the left hand was much larger in violin players than individuals that did not play the violin. So just like they observed in the owl monkey study, there was compensation in the brain, plasticity in the adult brain following stimulation. So when we age, right, as we go through ages 50 and onward, the adult brain goes through changes where many sensory abilities decrease, unfortunately. This is something for you to look forward to and for me to experience right now. So vision and hearing decline, unsurprisingly. Some scientists think that, okay, maybe the sensory neurons in these sense organs are deteriorating, but it could also be the neurons in the brain that handle those sensory functions are weakening. In addition to their sensory abilities declining, adults that age also lose cognitive abilities, and this is something even I've noticed. Most adults experience a reduction in processing speed. So what this means is the speed at which they're able to think about things and process information coming into their nervous system declines as they age. When scientists have taken a look at the brains of aging adults that have died, so post-mortem, they found that not only did the brain weight decrease, but the size of the neurons, as well as the connections between neurons, have all decreased with age. So what could be contributing to this decreased neuron size, decreased neuron amount, and decreased connections in the brain? There's one reason, although there might be many, but I'm going to share one reason with you why this might be the case. And it has to do with a genetic cause of an age-related change. Genetic decline in particular. So your chromosomes have kind of like a cap on the ends of them, and these caps are called telomeres. Telomeres serve kind of like the same purpose that a hem does on a shirt or a skirt. The hem prevents the fraying of the fabric over time, right? And so the telomeres, these caps on the ends of your chromosomes, serve the same purpose. They protect the DNA as it undergoes replication in our bodies. But as we age, these telomeres shrink. They get thinner and smaller. And so as a result, they don't do as good a job at protecting the chromosomes from damage as they used to. When a cell, a neuron too, when a neuron experiences damage in their DNA, that triggers the cell death response, right? So it doesn't want to keep around a cell that has damaged DNA because it's not going to be able to produce functional proteins. And so it initiates cell death. And so if we've got lots of neurons whose telomeres are shrinking, whose DNA is at greater risk for damage, those neurons are going to die. And if we have many, many neurons that are dying in the aging brain, that could potentially lead to cognitive decline, right? The neurons that were handling the cognitive function are dying as a result of the normal aging process, but that may be one reason what is contributing to cognitive decline. An aging brain will experience normal cell loss. But there are diseases of the aging brain, like Alzheimer's disease, where the loss is much, much more dramatic, right? It's not normal. The pace at which cell loss occurs in diseases like Alzheimer's disease. You may or may not be familiar with this, but Alzheimer's disease is a type of dementia. Dementia refers to disorders of the brain where there's massive cognitive decline. There are many different types of dementia, and Alzheimer's is one type of dementia. What's often characteristic of Alzheimer's disease is shrinkage of areas like the hippocampus, enlarged ventricles and spaces of the brain. The cortex is also dramatically shrunk. So what is contributing to this shrinking? And what makes Alzheimer's disease different from other forms of dementia are two very important characteristics that are only found in a brain that has Alzheimer's disease. And now scientists have actually figured out a way to measure these two, or at least one of the characteristics, in a living brain. Prior to this, like 10 years ago, the only way they could verify that somebody has Alzheimer's disease is after the person had died, because what they have to look for in an Alzheimer's diseased brain is the following: amyloid plaques, which are typically found on extracellularly, like outside the neuron, and neurofibrillary tangles. So we'll talk about neurofibrillary tangles in a second, but amyloid plaques are they look like hardened tissue surrounding the outside of the neurons. Specifically, they might get in the way of functioning between neurons, right? So you might see amyloid plaques between the terminal of a neuron and the dendrite of, or the surface of another, preventing this neuron from communicating with each other. These amyloid plaques are kind of like, blockages. They prevent neurons from functioning normally, and you can imagine a neuron that's not functioning properly will eventually die and not stick around. So amyloid plaques is one of the characteristics of Alzheimer's disease. The other is neurofibrillary tangles. So one of the things that are present in the axons are these kind of railroad cytoskeletal proteins called microtubules. And what microtubules have on them that stabilizes them are these Y-shaped tau proteins. These tau proteins exist on microtubules to give them structural support. For some reason in Alzheimer's disease, these tau proteins pop off the microtubules, making them destabilized. So not only do these microtubules stop kind of keeping their shape, but they also fall apart. And these tau proteins bunch together to form what's called a neurofibrillary tangle. If you look at the cell body of an Alzheimer's diseased neuron, you will see that it's completely clotted with a neurofibrillary tangle. It's almost like, a hair clog in a drain. If a cell does not have the ability to move proteins and move its organelles in its cell body, it will not be able to function. And that is definitely the case with cells that are clotted with neurofibrillary tangles. They can't function if they can't function, they end up dying. And so the two characteristics of Alzheimer's disease are amyloid plaques and neurofibrillary tangles. We still have to look for neurofibrillary tangles post-mortem, but now scientists and doctors have figured out a way to measure amyloid in the brain so that they can verify that a person has Alzheimer's disease before death. So these types of changes that occur not just with Alzheimer's disease, but rather with the normal aging process is called negative plastic changes. So these are changes that go against changes that are beneficial to the brain, right? Typically, plastic changes are the ones that are changes that are helping brain function. Negative plastic changes are those that occur where the brain is losing connections or losing neurons. So the signal to noise ratio gets worse. What that means is our ability to extract meaningful information from our environment decreases because there's so much noise and clutter in the normal aging brain. So as this worsens, our brain is not able to function as efficiently as it used to when we were younger. But there are plastic-based training programs that people can engage in as they age to counteract these negative plastic changes. So they can work to enhance their sensory functions, their processing speed, and even their memory. And there's some research to suggest that when people engage in these plastic-based training programs as they age, their performance actually increases in these areas. And as I alluded to earlier, I want to share with you the fact that exercise can also improve brain plasticity and function in aging. So I'm going to share with you a study that's a little bit old to date, but it is super interesting still. It's an experiment that was examining the role of aerobic exercise specifically on hippocampal volume and memory function. So they took a bunch of aging adults, anywhere from like 55 to 80. There were 120 adults, and they split them, or they measured them for brain size, memory function, fitness, and they measured different chemicals in their blood. Then they split these 120 subjects into two groups. One group engaged in aerobic exercise, and another group, for that same amount of time, engaged in stretching, kind of like, not yoga, but kind of like yoga. And I should mention that both the aerobic exercise and stretching occurred in the laboratory. So they came every time they engaged in these behaviors or activities. At the 6-month period, participants in both groups were measured for their MRI, for their brain volume, their memory function, as well as their fitness. And after 1 year, they were measured for all of the characteristics they were originally measured at baseline. So here are the results from the MRI portion of the study. So they were measuring the size of the hippocampus, and what I've delineated here in yellow is where the hippocampus would be in the adult brain. And they were measuring the volume of both the left and right hippocampus, because you have one on each side. The exercise group is in blue and the stretching group is in red. So at baseline, both groups were nearly identical for their left hippocampus, and it seemed like the exercise group had a larger right hippocampus compared to the stretching group, but that was completely random. Here's what's really interesting is that over the 1 year period, the exercise group increased in volume in both their left and right hippocampus compared to the stretching group. Okay, so this suggests that engaging in aerobic activity can increase structures of your brain, right? What they later found was that the exercise group that had their bloods measured, they were making more of the neurotrophic factor BDNF. So BDNF causes exercise causes increased production of BDNF, and BDNF, as you know, is really important for neuronal survival. And so these participants that were in the exercise group had more BDNF, which likely contributed to the larger volume in their hippocampus 1 year after starting the study. What's really interesting is when they did a correlation between the right hippocampal volume and the memory performance, they found that participants who showed an increase in their hippocampal volume also showed an increase in their memory performance. So there was a significant positive correlation between the size of the hippocampus and their memory function capability. So this suggests that exercise not only increased the hippocampal volume, but that increase in volume resulted in improvement functionally because these participants also performed better on memory tasks. So this study I think is really interesting because it illustrates this idea that you don't need to engage in brain training games all the time. You need just some sort of stimulation, whether it's physical or mental, that's going to help combat the negative plastic changes that often accompany the normal part of aging. So we are done for this lecture. Just to summarize a few important points from this lecture, the human brain matures slowly and well into the second decade. As a result, the adolescent years have poor executive functions and are more likely to engage in risky behaviors. The adolescent years are also characterized by onset of several mental disorders, although it's not as plastic as what is true in development. The adult brain maintains neuroplasticity as the brain ages. There's reduction in sensory and cognitive abilities, and even greater incidence of diseases like Alzheimer's disease is present in the aging brain. It's possible that damaged telomeres are contributing to the loss of functioning with aging, but normal cognitive decline can be counteracted with brain training programs or even exercise.